welcome to our today's lecture, and I hope that you all know me by now, and if not, my name is Tatyana and I'm a PhD student in this lab of Professor Rost. And today I have the special pleasure of talking to you about a subject that I started to work with about 3 years ago as my master thesis project, and then 2 years ago it turned into my PhD project. So the topic is the development of a prediction tool for subcellular localization. But to begin with, let us answer the question of why do you want to predict the protein subcellular localization. So who knows what this is? A cell? And what kind of cell? Huh?
Lipidic.
What?
Lipid.
Lipidic.
Yeah.
What is that?
The circle.
Yeah, there are lipids on the membrane, that is correct. But there are...
Some yeasts or
well, there are bacterial cells, there are eukaryotic cells, animal cells, plant cells. What kind of cell is this? Do you know? Do you know?
eukaryotic cell.
Yes, what's a eukaryotic cell and how do you see it?
Because it has a nucleus. So
A nucleus, exactly. And plant cells, for example, they have an additional cell wall on the outside, and they are usually not this, they don't have shape like this as animal cells. And what are these parts in the cells? Like these colored things, do you know?
the blue color or
Yeah, blue colored, white colored, pink colored, green colored. This here, what is this?
some proteins. The blue is mitochondria.
Organelles.
Oh,
Organelles, like mitochondria, like this here. Which else do you know?
energy.
They produce energy. Golgi here, yes. What else? Can you name 5? So we have 2. You said nucleus before. You are very close with the lipids, that is the membrane, which is also an organelle. A compartment, it's not an organelle. Organelle is something that is enclosed by a membrane. So this is compartment. ER. Exactly, and the plasmic reticulum, this here. Yeah. And in each of these compartments we have thousands and thousands of proteins, and depending on the compartment, the function of a protein is different. So it's obvious that protein that sits in the nucleus would have a different function than the protein that sits, say, in the mitochondria. And here I want to show you some examples of proteins and their functions. Do you know what a nucleosome is? Have you talked already about it? Okay, so this is a kind of protein that sits in the nucleus of the cell, and it packs the DNA to make it compact. So this is the protein, this is the DNA. It packs it to make it compact, and it also protects it from damage. Ribosome. What is a ribosome? Do you know? So this is a kind of protein that you can find in the cytoplasm or in mitochondria, and this protein is involved in the synthesis of other proteins. And humans, eukaryotes, bacteria, they all have ribosomes, and for example, antibiotic drugs, they target specifically bacterial ribosomes, because we don't want bacteria to replicate to produce new proteins. But luckily for us, our ribosomes are very different from bacterial ribosomes, and that's why when we take antibiotica, then our ribosomes are not being affected, only bacterial ribosomes. Collagen, that's another very interesting protein, and about 1/4 of all our proteins in our body are collagen. And these proteins, they support our skin and our muscles, and our bones and teeth, they're also made by this protein by adding minerals to them. And these proteins we can find outside of the cell. Crystalline. So this protein we can find in the eyes, in our eyes. And this protein, it captures the light and it allows us to see. And interestingly enough, when we are being developed at very, very early stages of our life, these cells in our eyes, the lenses, they get filled themselves with these proteins, and then these proteins, they stay for the rest of our life in our eyes. Interesting, I think.
Is it decreasing or stays the same?
It's decreasing with the life, yeah. And sometimes the eyes of older people, they're not as transparent, and that's because these proteins, they break in their structure.
So can you just
fix it by putting new crystalline inside?
That could have been idea.
So interestingly, the cells of our eyes, they don't have nucleus, so they don't have DNA, and that's why new proteins cannot be produced.
Okay.
So the cells, they sacrifice their DNA for these proteins. ATP synthase is an extremely cool protein. And this protein can be found in the membrane of mitochondria, and they produce energy. So what happens here, so this is the membrane of mitochondria, and what happens here is that hydrogen ions, they flow through the membrane, and because of this flow, this part, it's a motor. It starts to rotate. And this part is connected to the bottom part, which is a generator. And when this one rotates, the bottom one starts also to rotate. And because of the rotation, it produces energy in form of ATP. So what happens when we eat, the food is being broken down to particles, and these particles make the ions go through. And because of the flow, this thing rotates, makes rotate this thing, and then the energy is being produced here on the bottom. So that's how it works. And another extremely important and also I think very cool protein is G protein. And G protein sits in the membrane of our cells. And what it does, so all cells, they communicate with each other, right? And the communication is made by messengers, by specific messages. And the messages can be proteins or they can be hormones, like adrenaline, right? That makes us be excited, or dopamine, or you name it. What else is there? What makes us sleep? Okay, I don't recall it right now either, but yes, exactly, these kind of things. And they're being transported from a cell to a cell. And these proteins, they sit in the membrane of the cells and they capture, they see these messengers, they recognize them, and then these messengers, they do not go through the cell, they don't go into the cell, but this protein activates some processes in the cell, and so the cell knows that it has to react to the thing somehow. So some drugs, many drugs, they actually attack this kind of proteins, and also drugs of abuse like marijuana or cocaine and so on, cigarettes. They also attack these proteins. And for example, there is...
How much is the level of attack?
Sorry?
In terms of marijuana, how much is the level of attack in this program?
It's not the level of attack, it's the level of how long a substrate is going to sit on this protein and block it.
So does it completely destroy it, or?
No, it blocks it. It blocks it, and then this protein cannot transmit anything to inside the cell.
Okay.
So yeah, the cell is blocked from the outside of its world. Nerve cells communicate to each other just like that. They send particles from one cell to another. So for example, when we have headache, that's exactly what happens. And then we take some aspirin that blocks these receptors, and then we don't have headache anymore. And there are specific bacteria that what they do, they produce special particles that again bind on these proteins and make them active all the time. And then you can imagine if the cells are active all the time, they always expect something, then the processes in the cell, they're corrupt. And that's why people get sick. So yes, any aspect of the role of a protein in a cell is called protein function. And it is one of the major challenges in the modern biology to understand what the function of a protein is. And I want to give you an example here. Who knows what UniProt is? Yes, what is it?
It's a database for
Oh.
proteins. So protein structure and protein sequences and getting to know proteins.
Of protein sequences and their annotations. Structures there in PDB. And what is SwissProt? And what is it? Do you know, David?
I think it's similar to UniProt, but I think it's people manually annotate.
Yes, almost correct. So UniProt, this is a database of all known protein sequences that we have as of today. So when we sequence a new organism, then the sequences, predicted sequences, we predict the genes, the proteins, and then we store them in UniProt. And then UniProt consists of two parts, called Trumbull and called SwissProt. So So Trumbull contains, we sequence an organism and we put all predicted proteins in Trumbull. So we think these proteins exist. And then biologists come and then they make experiments. And then if they see this protein exists for real, then they make an entry in SwissProt. So they take the sequence from Trumbull and put in SwissProt. So SwissProt has all manually annotated proteins. And so we can see as of yesterday, the UniProt database contains 87 million protein sequences, which is a huge, huge number. And it's actually great to have this number because this many protein sequences, they help us to understand all these complex biological processes that take in the cell. They help us to understand the differences between different cell types and diseases. But if we look at the number of proteins in SwissProt, we see the difference is significant. It's really, really big. And so it's obvious that there is a need for some reliable automatic prediction methods for protein function from amino acid sequence alone, because manually it's absolutely not possible to handle this number because, yes, you see how big it is. So what we want in the end is something like this, where we develop a predictor that takes a protein sequence as input and outputs a protein function. And this is actually what my lecture is about the development of a prediction method like this. A function of a protein can be described in many, many different ways. So it can be described on a physiological level, on biochemical level, on chemical level, and so on. So a description of protein function can be very, very confusing. But when we develop a prediction method, we want to have a somewhat organized vocabulary that the computer method can handle with and can read and understand. And so gene ontology, this is a vocabulary that tries to organize protein function somehow, and this is what came out of it. So gene ontology is a hierarchical organization of terms that describe protein function, and gene ontology is divided into three classes: cellular component, biological process, and molecular function. So every protein can be described by terms that belong to one of the three classes. What do you think? What kind of terms, what kind of things can be described by molecular function? So what is a molecular function of a protein? Can you give an example? Like what can be a function of a protein?
What they produce?
What do they produce? What proteins produce? Other proteins. Synthesize new proteins, yes. What else?
Supporting the skin, like you told before, the collagen, isn't that a function?
Yes. Yes. They bind.
Binding oxygen.
Binding, holding, moving, transporting, modifying one thing into another thing. So these are all molecular function terms. And what could be a biological process? What could you think of that could be part of a biological process? What is a biological process?
Production of ATP. Production of ATP.
Production, yes. What else? Okay, what have you learned in this lecture so far? What is a process? Give an example of a process. Synthesis. Translation. Transcription. Cell development is a process. Can you think of a different?
Metabolism.
Metabolism is a process. Mitosis is a very good process, cell division. Yes, apoptosis. So these are all biological processes. And here I put the numbers to show you how many terms we have to describe each of these two classes. And then there's also cellular component. And this is exactly the localization in the cell. These were exactly those colored organelles I was showing you before. And what do you think? How many terms are there in this vocabulary that can describe cellular component? Can you give an estimate? A rough estimate.
Thousands?
Like how many? Like 2,000 or 200,000? 2,000? Okay, well, not bad. Kind of close. But that's impressive, I think. And this here is an example of a cellular component ontology for a human protein histidine. So this protein is found in the nucleus. You could say it's found in the nucleus, but gene ontology would show you a tree like this.
All those terms in Homology are manually annotated, right? So
Yeah.
somebody thought about it and okay, it's
And
about 3,
put them into this hierarchical structure, yeah.
Okay, so how can you be sure you get everything?
What do you mean?
So maybe you missed something in the cellular component and somebody think about it?
Well, if it's not in there, then you define a new term as well. Yeah, that's how it works. So yeah, proteins, they can have many different functions, and this here is some biological background. Okay, so what are these two?
Prokaryote and eukaryote.
Very good. So all living organisms, they can be divided into prokaryotes, which are bacteria or archaea, or eukaryotes. And the defining feature of a eukaryotic cell is the presence of the nucleus and other membrane surrounded organelles. And yes, each of these compartments contains, as I said before, thousands of proteins. And we believe that if we find two proteins in the same compartment, say in the nucleus, then they will probably have a more common function than two proteins that one sits here and the other sits, say, in mitochondria. And so our conclusion is that, well, common compartment defines, somehow leads to a common physiological function. So we think that if proteins are localized in the same compartment, they will probably have the same function. And so this is the idea that drove the development of all actually localization prediction methods. And one application of the knowledge of subcellular localization is in drug discovery. So this here is a chart that shows the composition of localizations of proteins that are targeted by drugs. Okay, so medical drugs target proteins, and this is where we can find them. And we can see that more than half of these proteins, they're localized in the membrane. Other popular compartments are extracellular space and cytoplasm, while the interior of the cell is not so popular to be targeted by drugs. And can someone explain me why is this the case? Why do we have this extreme distribution?
If you don't have the virus or bacteria in it, the cell won't be contaminated.
Okay, say it again.
If the membrane is the target, it's because if you don't let the virus or bacteria go inside it, block it, the entrance, the cell won't be affected.
The questions would be different. So this is a human cell, okay? And then there are everywhere proteins. And then you develop a drug. And then the drug targets specific proteins. And here we see that most of the proteins, they would sit in the membrane. And my question is, why is it the case? Why do you want to target proteins mostly that sit in the membrane, in the cytoplasm, or in the extracellular space, not in the cell at all? So you as a biologist, developer of chemical drugs, why would you be interested in this kind of proteins especially?
Because maybe you can control what, this way you can control what goes inside and outside of the cell.
Yeah. Maybe other reasons?
Maybe it's easier to target them when they're sitting in the membrane.
Exactly, yeah. So you don't need to think of any construct that would make your drug go into the cell. Well, because you can simply find these proteins that already are so close, are in the membrane or extracellular space. So this is the only reason actually. And another reason is again this G-coupled protein receptors that are already in the membrane. And so this is why you want to target them. Okay, and this here is a table that defines what is a good drug target. So if you take a protein and then you look at different features, these features, then they will tell you whether a protein is a good drug target or not. And so we can see that, for example, membrane location is a very, very important feature for a protein to be a good drug target. Okay, and to summarize, drug targets, they tend to be found in membrane, cytoplasm, or extracellular space. All right. this here is another table that shows you subcellular locations, drugs, what they do to proteins, and when we use them, when we apply them. So I don't want to go through this table, but this is just interesting. It's nothing for the exam, but if you're interested, you may go through this table when you're at home. Okay, and another, I think, very interesting application to know about the knowledge of subcellular localization, and that's in mislocalizations that are associated with human diseases. So, for example, if we look at this protein, and this protein is normally localized in the nucleus, but if there is a mutation in its localization signal, then we know that there will be a loss of nuclear localization, and this can lead to a disease. Okay, I don't know what this particular disease is, but from this table I can see that mislocalizations, they may lead to really, really bad diseases like different types of cancer, or, okay, well, this is the only one I can recognize here, like speech language disorder.
I have a question on So the protein is produced with the ribosome outside the nucleus, but it's not already produced inside ventrally, right? Mm-hmm. So it has to find a way somehow inside the organelle or membrane. How does it test this GPS inside? Okay,
I will come to it in a few minutes. That's very interesting. Thanks for paying attention. So the message here is that something that leads to mislocalization of a protein may lead to diseases. And this here is an example for that. So what is this? Who knows? What do we see here?
Synapses.
Synapses. I mean, the whole figure, what does it show?
Mitosis, maybe?
I rather see 2 cells. Okay, 2 healthy cells. Okay, so this is the nucleus, and this is somehow the cell. And here, on this picture, I do see, you just said, someone just said?
Mitosis.
Mitosis, cell division. And this is how healthy cell division looks like. So you have here this spindle apparatus, and you have this microtubules, and then they push one cell into 2. The DNA of one cell into 2 cells. Okay, this is what happens normally. Okay, and this is apparently not the healthy state, and what's wrong here? We can identify something here.
Looks like exploded.
Yes, so what happens here is that a protein called protein kinase C loses its localization. So normally it lives in these organelles called centrosomes. So it loses its localization from there. And then what happens is that the cell is not able anymore to divide. So this is what happens. It tries to divide, but it's not being able to divide, and then you get a cell with 2 nucleus. And then what happens next is that it tries to divide again. And then there are more than 2 cells. And this tries to divide again, and so on. And this is what happens in the end. And what is this? Do you know? Cancer, exactly. So mislocalization of one single protein may lead to something like this. So this is another heavy reason to know what the localization.
What leads to mislocalization?
Mutations, for example. And when mutations happen, well, this is a different subject on its own. Yeah, well, why can mutations happen?
Environment, some influence from the outside.
Exactly, yeah.
Radiation.
Yeah, exactly, yeah. So normally mutations, they happen all the time in our bodies. But our bodies, they're very, very clever. So usually they recognize these mutations and then they fix them. But sometimes when we're exposed to something, yeah, the system is not that stable anymore, and mutations, they happen to occur. Yep. Exactly, so the scientific name for it is cytokinesis, the cell division fails. Okay, so why do we want to know the subcellular localization? Well, because knowledge of localization provides information about its functional role. So if we know a protein sits in the nucleus, we can assume it has to do something with the DNA. Well, the subcellular localization helps us to improve target identification during the drug discovery process. Aberrant, mislocalized proteins, they have been observed in cells of several diseases. And finally, knowledge of localization can help us to validate or analyze protein-protein interactions. And why is this the case? Why localization helps us to analyze protein-protein interactions? So what are protein-protein interactions? Okay, protein interaction. What happens here?
Proteins.
Interact. Exactly.
They have a coffee.
They meet. Exactly. And why localization is important to know whether this protein-protein interaction happens or not.
They can only interact when they are locally close to each other.
Exactly. When they sit in the same compartment or maybe in neighboring compartments. There you go. Okay. Well, one way to determine the localization is to use experimental methods. Like, for example, shown on this figure here, this is a fluorescent image of 2 cells. And these experimental methods, they are effective, they are kind of fast, but they cost money, they are not accurate, and most importantly, they are not complete, because mostly biologists, they're either interested in a specific species, like for example human or a mouse, or in specific organelles, like for example chloroplasts in plants. And in everything else, they're just not interested, they don't make the experiments. And for example, an extreme example, so this is, who knows who this guy is? What is this? This is a scientific name for whom?
Sugar?
Sugar something is not an organism. But it has to do something with sugar, yes. David, do you know?
No.
Oh my god, guys. So this is yeast. This is a scientific name for yeast. So yeast has been studied for many years now. And there have been so many experiments done to determine the localization of proteins. And if right now we have so many manually annotated entries in SwissProt, so we know that yeast has so many proteins, then what we will find right now is that only 2/3 of these proteins, they have a localization. Meaning that 1/3 of their proteins, they still have no localization information determined experimentally, even though people work on it now for decades. And that's just kind of crazy. And so you can imagine that for other organisms, take, I don't know, well, whatever is out there, a bear, a tiger, a cat, a dog. This percentage will be much lower. And so it's obvious that we do need computational methods that predict this localization for us. Okay, and the localization is, a matter of fact, a very nice playground to develop a prediction method. And in predict protein classes like this here before, we were actually working on the development of prediction methods, including localization. And actually, we have even published one of these methods developed with our students. And one of the reason why localization is so nice for method development is because localization is somewhat easy to define. So if we see that protein is localized in an experiment in the nucleus, then we can be sure it's not localized, say, in the ER. Well, another reason for localization being so nice to predict is that the process of protein trafficking, of how a protein moves from one compartment to another, is very well understood. Another reason is that, well, we have kind of a lot localization data in our public databases, so we can develop these methods. And one last reason that I want to name here is that, well, even though proteins, most of them are being synthesized in the cytoplasm here, they, to get into other compartments, they need to travel sometimes to other compartments. So they change their localization, but still we think that they function in the very end, when they reach their final compartment, that they function in only one compartment. And this is the compartment that we want to predict. And Yes, as I said before, the protein trafficking in the cell is very well understood. Okay, so what happens is that most proteins, so we have nucleus, we have the DNA, right? And then the synthesis of a protein happens in the cytosol. In mitochondria, it also happens in the mitochondrial cytosol. And then this protein needs to be brought to different compartments. Okay, and then there are different pathways. Actually, there are three main pathways of how this happens. So one of them, this is gated transport. So proteins, they move between cytosol and the nucleus, there and back. So there are special pores, and they just go through the pores there and back. Or sometimes they just stay. This is the whole trafficking procedure. Some other proteins, they use the transmembrane transport, and they go to these compartments through the membrane of these organelles, and then there's also this vesicular transport. And what happens here is that the protein uses a special vesicle that it uses as a highway to travel to different compartments. So for a protein to get to cell outside, it has first to go to the yard and to Golgi, then maybe to here, and then to here, or straight to here. And so we kind of really know how proteins, they travel in our cells. And the trafficking is determined by specific signals in the protein sequences. And the signals we call sorting signals. Can you explain this figure for us? Because I see that you're falling asleep. So what kind of... Can you explain to us what kind of sorting signals are there? So based on this picture. So there are two types of sorting signals. Can you explain what are they?
I don't know.
I'll try. So this is very basic biology. If you have understood this, you already won. Okay, so...
I remember this picture already.
Oh, you do?
So already showed us this picture.
Oh, there you go. So then you even
But I actually
repeat. Okay, so let's try to figure it out together then. So these are protein sequences. And it starts here and goes to here. This is the beginning, this is the end. Okay, and then the signals, they are shown by these red stripes. This is one kind of signal, and this is the other kind of signal, sorting signal. So what's the difference between these two?
One is only on one edge, and the other one is distributed along the whole length, and the red parts come together and give the structure of the protein.
Very close. Okay, so one signal is called signal peptide. You can find it on either one of the ends of the protein sequence, just as we said. And the other one is distributed across the sequence, and it forms a signal only then when a protein makes its three-dimensional structure, and then they come together and they form a signal. So what happens is that so a protein has this signal, okay? And the signal tells some other protein, a transporter, where to bring this protein, to which compartment. So what happens is that another protein binds, he sees the signal, this tells him, okay, I need to go to, say, ER, and then he brings him to this compartment. And in this case, usually this part is then being cleaved off, it's being cut off. The protein then lives without this part. Yeah, in this case it does live with it, because we cannot cut off. But this is the mode of how proteins travel compartments.
But in the second one, the signal works only when the protein is folded.
Yeah.
Okay. Because it has to like touch or.
Yes, they have to come together in the 3D structure. Like this, they don't touch my fingers, but like this, they touch and they form a signal.
And it works.
So another protein can see it and bring. Yeah. So for this signal, the protein does need to fold, but for this one, it doesn't need to fold.
But I still don't get it. What is the signal? It produces something or not? Or is there a part of the cell that connects to the external world?
Okay, so this is a protein sequence, right? So these are amino acids like A, W, H, M, A, D, whatever amino acids are there. And the first, say 10, they form a signal. So some other protein will come, will see these 10 amino acids and will know that they're specifically responsible for localization of mitochondria. Say. And then this protein will be brought into the mitochondria.
Okay, so there is another protein that looks at a H2M and they say, okay, I can connect it here and it connects.
Exactly. Yeah. And so the whole project, the whole talk is about predicting protein localization, right? So if we would know all the signals, the prediction problem would be solved. But the problem is that we do not know the signals. So we know only very, very few of the signals. People say less than 10% of these signals. And these signals, well, you can imagine they're very, very difficult to identify experimentally. So for this, we know even less. And as a matter of fact, there was a Nobel Prize given in 1999 to this professor, Günter Blobel is his name, for finding these specific signals that govern protein transport and localization in the cell. So he called the signals address tags or zip codes. Why is that? Because if you write a letter, okay, and then you say, I don't know, person Peter lives in St. Petersburg at this street. So I know that there is a St. Petersburg in Russia, but there is also St. Petersburg in USA. And then the postman, they wouldn't know where to deliver this letter. But if you put a zip code, then the postman will exactly know that this letter is addressed to a person in Russia, for example. And the same happens with this sorting signals. So the active zip codes, they are very, very specific for particular localizations.
So how does the postman protein know where to go?
Sorry?
So you have a signal which tells you where this protein should belong to, and then there's another protein pulling it to the location.
Exactly.
So how does the other protein know where to pull?
Well, they bind with each other. So if they manage to bind, then it's being transported. If there is no binding,
so when it's binding, then still it has to find its way into, let's say, the mitochondria. But how? So how through the cell?
Well, there is a specific transporter that is responsible for bringing proteins to mitochondria.
Okay.
Yeah. So that's how it works. Yeah.
You said that if we know everything about signals, there would be no problem for the localization prediction.
Exactly.
But of course, we don't have.
Yes. That's the tiny problem.
Just one more thing. If proteins are not close to each other, how they communicate with each other? Are they just traveling randomly around, and then when they found signals, then they know what to do, or how they define this movement?
Have you seen these pictures, these videos from, say, New York, with the streets that are packed with people and they cross the streets? And they're like, all you see are just people. You know this, right? And that's how it looks in the cell as well. So the cells, they're extremely crowded with proteins. Like actually they all touch each other all the time. Yeah.
So basically, it's random interaction. They don't know who will they meet next, which is the protein, but they just go with the flow, and then when they find some signals, they bind together?
Yeah.
Is that the way?
Yeah. But it's a clever way. I mean, the evolution has been working on it for millions of years, so it happens so efficiently. If there is a mutation, then there is a problem. Then they cannot bind each other. But normally, yeah, that's how it goes. Okay, so interestingly, there was a Nobel Prize given in 1999, and just last year there was again a Nobel Prize given just for a very similar discovery, and the discovery was for the discovery of the machinery that regulates vesicle traffic, a major transport system in our cells. So you see that this localization problem is still something that makes people occupied, and is still something that they give Nobel Prizes for. And I think this is extremely cool. And as a matter of fact, Randy Schleckman, he will be in Munich in about a month, and he will be talking about this, about the localization of proteins.
In 2017, maybe you'll be...
Can you say it to the camera?
What is, I mean, maybe I'm not that much understanding basic things, but what is the implication, like, that had application in, like, real, what has been done yet, like in cancer or something like this, due to this localization, what can be avoided?
Anyone?
That's the question.
Yeah.
Okay, can other people answer this question maybe?
We said before, we use the localization because we know better which functions
Why are we interested in the functions?
Better target with drugs?
Yes, one application. What else?
And you can also target the pathway. So if you know how this works, then you can specifically target the pathway to change some processes.
Yeah. Have you heard maybe of some plants that are more resistant to cold or something?
OGM.
Or that. And that's exactly what's happening. So we target specific proteins to improve the survival of a plant, say.
What about for humans?
For humans, so an example when I was talking about ribosomes, so you want to target what?
X-Men.
So you want to target bacterial ribosomes, for example, with antibiotics. So you don't want them to replicate, you don't want them to reproduce themselves, you want them to die. And that's what you do, you develop drugs that target specific those proteins, bacterial proteins that are responsible for their survival. Same happens with viruses. Yeah.
Can you use this to correct mutations? For example, I have a cell that's mutated, and they're giving the wrong signal, so I create another protein that gets that signal and gives the right one, like good.
Yeah, that's what happens, yes, now as well. So you have the signals and then you attach them to other proteins that you want to bring into the nucleus. And then you attach it and the protein goes into the nucleus and performs its function. So bacterial proteins. So some proteins, they're really nasty. Some bacteria, they're really nasty. For example, chlamydia, maybe you heard about it. So what happens there is that, so a bacterial cell produces protein, and then it shoots its protein out of the cell into a eukaryotic cell. And the thing is that this bacterial cell has a signal that looks just like a human nuclear localization signal. And the human cell thinks, oh, I know the signal, let's bring it to the nucleus.
Okay.
And then this is exactly what happens. So bacterial protein gets into human nucleus and does stuff. The person gets sick. And so you do want to know the signals and you do want to identify what these proteins go to. Any other questions?
I'm just wondering, is it possible then to change, let's say, because we are hearing a lot now that they're trying to improve some structures of DNA to bring some new features or to improve some features. I'm wondering if this is possible already with like when humans while they are grown up already, or this is possible only while they are in development, like when they're really like I don't know when they're developing, when they're still not born or something.
Well, okay, one thing is that on humans you would not make this experiment as of right now. It's not allowed. But on mice you do it from the beginning before, yes. So a cell needs to divide in order to propagate its gene.
Okay, so basically very early.
If you work with the DNA, yes, from the very beginning. But if you work with proteins, again, you can eat a drug and then it will find its way in the diseased cell. So yes, actually to the main aspect of this whole lecture, predict localization. So what are the ways of how we can predict the localization? Okay, one of them is to find the sorting signals, but as we said before, we don't know as many sorting signals. Another way is to do homology-based inference. And what is this? What does it mean? What is implied by the homology-based inference? I'm sure you have talked about BLAST, right? About homology. Yeah. Yeah. So how can we use homology to predict localization?
Probably proteins that have similar functions have also similar
Sequence. Yeah. I would say another way around. Proteins that have similar sequence, they will have similar functions. Because this is what we're interested in. Yes, exactly. So if we know that chicken, mouse, human, orangutan proteins, they're localized in the nucleus, then we can say, okay, then probably a dog protein will also be localized in the nucleus. Another approach is text-based analysis. So what you do here, you either read PubMed, which is a database of all publications, or you search for special keywords in SwissProt, and then based on this information you can say, okay, probably if these guys say that a protein is involved in transcription, then probably it will be localized in the nucleus. So this is how this type of method works. De novo, what is de novo? Again? No.
Without knowing anything before.
Huh?
So de novo is without knowing anything. Just like
From first principles. Yeah. So de novo, this is the kind of method. This is where machine learning falls into. Do you know what machine learning is?
Yeah.
What is it?
You create models to identify clusters, and you can make an automatic learning based on that field.
Yeah. So this is what happened here. So my method was a de novo method. What happened here is that it was learning the subcellular localization based from the amino acid sequence alone. So there was no other information necessary but the amino acid sequence. And then based on these sequences, my tool was able to predict the localization. So it was a de novo based method. And then there are also hybrid approaches, and hybrid approaches, they're just a combination of any of these four. Okay, before you start to develop any method to predict localization, you have to consider some problems. You need to be aware that there will be problems when you develop. And one of the major problems, I would say, is that the biological data is noisy. And the noise can arise at biological, experimental, or curation levels. So what do you think? What is the problem at the biological level? Where can mistakes happen or errors happen on the biological level?
I don't know where, but the problem for most is that the data is enormous. It's a lot of data. So
Yeah. Okay, but if you're interested in one protein and you spend 5 years on working on this one protein, and you perform an experiment, and you still have only one protein, but there can still be a mistake at the biological level. And what it could be? Huh?
Communications?
I think simpler. So you make an experiment. You're a biologist, and you sit at your how's it called?
You didn't saw something or
Microscope. How was it? Yeah, exactly. So what can happen is that something goes wrong with your experiment, and you simply make a wrong observation. You know this happen.
While they are doing this, then it is costly and there's high chance of mistake.
Okay.
They are spending money in this.
Okay, I will tell you a story. So once I was doing an internship in Israel, and they told me a story that a group has published a biological paper, and then another group in some other country was trying to replicate the experiment, and they were trying, but they failed. And they were like, what is it that does not allow us to repeat the experiment that we get the same results? And they did everything correct. Everything was the same. And what happened in the end is that, the reason was the room temperature. Because in Israel they turn AC on, and then it's very cold. And something like this can also lead to different results. Okay, another reason is experimental. Okay, what is this? So where can mistake happen at the experimental level?
You make a mistake during the experiment.
Yeah, something goes wrong with an experiment, exactly, yeah. And at the curation level, so curation, this is when you write down your observation or enter your data into the database, so what can happen here wrong?
Mistypes.
Exactly. So you made an experiment, you made the correct observation, everything was right, and then you give your data, you publish the paper, and then the person who puts your data into the database just makes a mistake.
The wrong file, mistypes.
Exactly, and then you have a noise, then you have a mistake in the database. So not everything that is experimentally defined is not 100% correct, so there can be errors even on that. And this is something that we have to cope with. So we cannot develop a prediction method that is 100% accurate. If someone tells you, okay, I have solved the problem, and prediction is 100%, then you will say, okay, this is bullshit, this is impossible. And this is the reason why. Okay, some other problem you have to consider when you develop a method is that, well, prediction performance has to be established properly. Sequence similarity between training and test sets is incredibly important. And why is that? So you develop a prediction method, machine learning method. And why is it important to pay attention at sequence similarity between training and test?
There's no similarity, it doesn't make sense. You can't learn actually. You can test on test data and get a good estimation of how good is your model.
So you want to have similarity or not?
You must have a similar, you must know what you have there.
Okay, but then if you learn, this is a chair, and then this is your training data, and then you see this, and this is exactly the same. So this is no learning, this is just, so this is not predicting, this is just learning by heart, right?
In any case, I think it's learning by heart. I mean, it's not.
But then it's not prediction.
At some point, at really low level, it is learning by heart actually. But I'm just wondering. What I want to say that in perfect case, you want to have a train and a test data, which are from different tissues, so not but the same sequence. And that would be the perfect data to do. I mean, I don't know if I use the right terms, tissues and so on, but usually we would use samples from different, let's say, objects and say, okay, now we know that these are same samples, but from different objects, and then you put this, split this in folds and try to test it.
Okay, so this is the big difference between computer science and bioinformatics. In bioinformatics, this is completely the other way around. So if you would have very, very similar sequences, then you would simply use BLAST, and BLAST would already find it for you. But why do you bother of developing a machine learning method? Because if you do not have any sequence similarity, there is definitely nothing similar if you compare them sequence with sequence. You still want to find some certain patterns in the sequence that would you be able to find in the other sequence that in general has nothing to do with it, but those patterns, they define the localization. So you do not want your data to be very similar. You want it to be very different. Because you want to extract from your training data those patterns that would define your localization. And if you see something absolutely new, then these patterns would be able to tell you that, okay, this is this localization or the other.
So every time you have a different similarity between training, so let's say I train my model with training data, which is similar to the test data.
Okay, so then this is learning by heart. You do not predict again. So you want them to be as different as possible because you want to extract those principles from the training data that would be applicable to something completely new. You sequence a new genome and then you want to know what is localization, and even if you have never seen the sequences before, you still want to be able to predict the localization.
I just think maybe we misunderstood some terms. The test data, is that the data that you're trying to validate your model?
Yes,
or
exactly.
the new data that you're trying to predict?
Both. So,
Indeed both. So
okay, when you develop your method, you train your model.
Okay.
And then you estimate the performance using the test data, right?
yes, that's like estimation.
Yeah, so and then you have your performance. And then you want to know, well, but how they perform, so this was my developmental data. And then you want to know, well, how they perform on something that is completely new, that I have not seen during development at all.
That's not the test data.
Independent
I would
test.
say that's new data set which we're trying to predict.
Exactly, but this is still test. You do testing, and you can estimate the performance of this data.
But the new test data set still has sequence similarity to the training data.
Well, you hope that it doesn't. You try not to have a similarity because you want to predict a absolutely new data set. You want to know how well you were able to train your model to identify those specific patterns. This is the question you're interested in. Yeah. Because predicting something very similar, the problem is very easy, so there is no challenge in it. But you want to know, you want to compete with other methods in predicting new proteins that no one has ever seen before. Yeah. Okay, So we clear that, but there is a problem, and that is that our databases are extremely sequence redundant. And I would ask you why this is the case, but I see there is no time, so I will tell you. And the reason for this redundancy is that, as I said before, people, they're usually interested in same species like human or mouse or yeast. And then if they perform an experiment for one certain protein, then they would also likely perform the same experiment for the same protein but from other species, because it's much easier, and usually this is what they're interested in a certain family of proteins. Okay, so we need to keep in mind that our databases are extremely sequence redundant. Okay, prediction results must be interpretable by users. So you just not tell them, okay, this is cytoplasm and that's it, live with it. You also want to tell them, okay, I think this is cytoplasm and this is the reliability score. If this is very high, then trust my prediction, but if this is low, then keep in mind that my prediction is probably crap. And finally, this is the benchmarking of your method with other methods, and this can be difficult. What you do when you develop a new method, you always compare yourself to the others. And why is this difficult? What is the difficulty in the comparison to other methods?
Bringing new methods can be difficult to compare to old methods.
Why? Yes, that was my question. So why?
Can you say that a prediction is better than the other?
How can I say? I calculate accuracy and coverage, and I say I'm better.
If there's like for some percentage, like 1-2%, how can you like convince the biologists to use your prediction instead of the one he's trained with and he's more used to use?
Yeah, this is one problem. But what are the other problems?
Benchmarking results, we need the same data set, we need the same setup.
Yeah.
Otherwise we can compare.
Yeah.
Basically, everything comes out from the data set that you have.
Yeah.
If it's not the same, so it can't be really benchmarked.
Exactly. Yeah. So it has to be independent for all methods. That's the new for all methods. And some other problem that I can think of is that, well, for example, my method predicts 18 localization classes for eukaryotes. But there is no other method, that, so far as I know, that can predict as many classes. So if I compare myself to the others, I have to reduce the number of prediction classes so that the estimate has to be as fair as possible. This is something. Okay, so if we keep all this in mind, we can start the development. And this is actually what came out of it. That was my master thesis called the Loc Tree 2 that predicts localizations for all domains of life. And so what happened here, I took the knowledge that we have as of right now about how proteins traffic in the cell, and I used this knowledge to develop my prediction method, and there was one system for eukaryotes, one for bacteria, and one for archaea. Okay, so this is some summary of the method. I will not go through this because this is not so relevant right now, but I want to talk about the development of this method. So the development involved 4 major steps: dataset preparation, training of the prediction method, testing of it, and then comparison to external tools. So again, if we would not have this idea of developing JavaScript visualization tools, this is exactly what you would have been done in this class. So the hardest part, this is to train the prediction method, because there are different machine learning methods, and each method works with different kernels, say, and so on and so on. They have their own parameters that you have to optimize. So this step is very, very heavy. But the first step was to prepare the data set. Okay, so for archaea, my data set had 3 classes, for bacteria 6, and for eukaryote 18. I extracted this data set from SwissProt, which is manually annotated part of UniProt, and this is how a SwissProt entry looks like. So this is a protein name, and this is a taxonomic classification. There's a line saying what is the localization, and there is a sequence itself. So that was just parsing of this kind of files. And then, as I said, there is always very strong bias in sequence redundancy in these databases, so we had to eliminate this bias. And one way of doing so is by using HSP value, and there will be one group, hopefully today, who will talk about what is HSP in the exercises, and by BLAST evaluate. So these are 2 criteria that actually eliminate sequence redundancy. So if you apply this to thresholds, then there will be no sequence redundancy in your data. Okay, and here is the statistics. So if this is the data I have extracted from Swissprot, then after sequence redundancy, this is how much there was left. So this is a very nice visualization to show how redundant actually is biological data. So this is something that we bioinformaticians work with. Okay, and once you have the data, you have to do a stratified k-fold cross-validation. So you need to split your data into folds. What for?
Validate, that's different performances if you take different stakes.
Mm-hmm. Can someone else try to explain why do you perform cross-validation on your data? Why do you have to split it? Why can't you just trace on the complete set of the data when you develop a method?
Because of the natural principle of divide and conquer.
What?
We have to modularize, divide.
Why? So why?
Can I consider that this data is filtered or not?
So my data was this big. For eukaryotes, for archaea, it was only this big. And still I had to divide, so it's not big. Still I had to divide into 5 parts. You can divide into 10 or into 3, I chose 5. What was the reason for doing so?
You can find blocks inside it, and you can compare afterwards with what you haven't trained, and then you go all around, and then for each block you have more consistency on finding what you want.
I think this is correct, but I would put a bit different. So if I would take this data and I would train on this one data set, okay, I would train a model, but then I would have no data to test it with, because we said training must be different from testing, right? And that's why I do this separation. So I do the separation here, did it in 5 folds. So that I would train the method on this part of the data, and then I would use the other part to test it. And then I would simply rotate. And then every single instance of my data would be tested at least just once. And then I would calculate the performance of my method simply by adding the values, the performances of these 5. And this is this is called cross-validation, and that's what we always do in bioinformatics when we develop our methods. Is that clear?
Yes.
Okay. Have you talked about it with Professor already?
No.
About machine learning in general?
In Homology one, but not in two.
Uh,
I think we went around this subject.
You did?
No, we will.
Ah, you will?
Yeah.
Okay, so then at least you will know what cross-validation is for us. Okay. So we used support vector machines to develop the method, and from support vector machines we actually know that we want to have as much data as possible, but my data was little, especially after redundancy reduction. So if this is my data after redundancy reduction, I wanted to increase the training set because I know that SVMs work better with the larger training set. So what I did, I took my whole Swissprot data from before redundancy reduction, and then around each training point I built a cluster of similar sequences, so that my training set increased, but there was still no sequence redundancy to the test data. And that was the trick, to make SVMs learn from even more data. Okay, but before we jump into the final performances, it's very important to mention what are the performance estimates, accuracy. Can someone tell what is accuracy in your own words?
Yeah, that's true positive divided by...
Come on, this is not your own words. This is reading from the formula.
Yeah, I'm trying to answer you want.
So how many actually got right?
Yeah, so if you have all your predictions, how many of those are correct? This is accuracy, and what is in coverage?
The amount of data you considered.
The tested one.
Yeah.
Yeah, exactly. So from all your instances that are belonging to this class, how many of these are true positives? Exactly. Accuracy and coverage.
Like test coverage.
Huh? What?
Like test coverage in software engineering.
recall sensitivity, or the other terms that I know. Okay, but then when you estimate accuracy and coverage, you still want to know that you are correct at estimating this accuracy and coverage. And then you want to add some value that would tell you how much you are correct. So if you would have, so on this data set, you have a performance of 65, and if you take another data set, would it still be 65, or would it be 66 or 67 or maybe 63? So how is this value called that you add to your performance value that Okay, and what is the, do you know maybe how is the procedure called to estimate the standard error? Huh?
Standard deviation?
Yeah, this is again the mathematical.
The difference of the squares.
Exactly, but where do you get all these differences from? So what do you need to do to get the values from other data sets? Okay, so look, you have your data set. You have accuracy for it and you have the coverage for it. And then you want to estimate the standard error. So what do you do? Because for the standard deviation you need other values, right? But where do you get them from? You only have one data set.
Like you divided the pie before?
Oh, yes, kind of. So I will get to this in a minute, but this procedure is called, in our world, like this. Anyone know what this is?
A dying horse.
By the way, does anyone know who this is?
Mislocalizations.
Shop.
No idea.
Alexander the Great.
I think.
Well, it's Munchausen.
Somebody who died. I would say Munchausen.
Very good. This is, and what is he doing here?
He's bootstrapping himself.
This is the part when he like pulls himself.
Yeah, so what happened is that he was riding his horse and he got into a swamp. And then the swamp was sucking him in, and there was no other help. So what he did, he pulled himself by his own bootstrap, and he pulled himself out together with the horse. So this is a true story, yes. According to him, this is a true story.
No, well, what? He took out the horse or?
No. He saved himself and the horse.
By pulling himself out from the swamp. So what happens here is that you do bootstrapping. You help yourself out when there is no other help. So yes, this term first appeared or was used in the adventures of Baron Münchhausen, but then it was also introduced by Bradley Epperlin this year in relation to machine learning, and it was popularized a bit later. Okay, so what happens here is that you start with your set of predictions. So you have a data set, and for each instance you have a prediction. Okay, and then what you do, you randomly draw a subset of this prediction. So if this is all your set, you take a subset randomly, and then for the subset you estimate the accuracy and coverage. And then you randomly take another subset, and then for this you estimate accuracy coverage. And then randomly you take another one, and another one, another one, another one, and so you get, say, 1,000 different values, 1,000 accuracies and coverages. And then from this huge set of accuracies and coverages, you can calculate your standard deviation and then the standard error.
Is it the mean of all the standard deviations? Could I say that?
This is your performance that you observed in your complete data set, and these are the performances from this bootstrap sets. Yeah. And this is standard deviation. Huh? Yep. So much to the statistics, and then there was the training of the method. Okay, so support vector machine, how they function will not be part of the exam, that's for sure. The SVMs, how they function, this will not be part of the exam. This we will not ask, but we can still go briefly through so that you know. Does anyone know what the SVMs are, how they work?
Yeah.
Do you want to explain based on this?
Basically, they draw a line that has the, let's say this is like a road, which is divided on left and right side, and they try to make the highest road possible to fit between two different clusters.
Classes, exactly,
Yes,
yeah, exactly. So what happens is that this is your data, and then you try to put a road in between this data that it separates the two classes as good as possible, so that the road, nice explanation by the way, is as wide as possible. And this road is called hyperplane. So the hyperplane with the maximum distance to both classes is the best one. However, most of our real data is not linearly separable. So most probably the data looks somehow like this, where you cannot draw a line. And so what you do then, you put this data into some higher dimensional space where you can draw a line. So say this is a 2D space, you put it into a 3D space, and then you can draw a line. And this transformation this is just an example, from 2D to 3D is made by a kernel function, and the transformation is called feature mapping. So a kernel function, this is what it does, okay? It transforms linearly non-separable data into linearly separable data.